Whole blood contains the formed elements of red blood cells, and hemoglobin is contained within those red blood cells. So, hemoglobin, as its name indicates, it's a globular protein instead of a fibrous one like keratin or collagen. And thinking back to protein structure, there were four levels of protein structure. So when a protein is first made from the DNA recipe, the protein is just a series of amino acids bonded together. That amino acid chain, it then becomes folded into alpha helices or beta pleated sheets. And then those helices or sheets become grouped together to make tertiary levels of protein structure. And then the tertiary levels come together to make a final quaternary structure that you see here in the call out from one red blood cell. We can measure how much hemoglobin is in your blood or in your red blood cells. This is what would be called the mean corpuscular hemoglobin, or the MCH, which is the average amount of hemoglobin in your erythrocytes. Anemia, such as iron deficient anemia, will have a low MCH. Hemoglobin actually contains two amino acid chains, one referred to as alpha and one referred to as beta. Whichever one you want to refer to as alpha and beta in my picture is your choice. These amino acid chains, the alpha and beta, they're tertiary structures. So these are tertiary or third level structures. The pictures that you see here are abbreviations of the tertiary structure because we know that it's made up of tons of little alpha helices and maybe some beta pleated sheets over there. When we take these two alpha chains and two beta chains, when we put them together, we start to see the final shape of hemoglobin. Bonded within the alpha and beta chains is a pigment called heme. It has usually a gray or a very dark silvery coloring, kind of like duct tape. This pigment, it contains another pigment called bilirubin. And bilirubin is the same yellowish pigment that gives stomach bile its characteristic yellow color. So at the center of all of this machinery are iron atoms. And these iron atoms, these are the components capable of binding to oxygen. There are four hemopigments, or four oxygen atoms, in every single hemoglobin protein. When oxygen attaches to the iron in the hemopigment, the pigment changes to a darker color. And this is the exact same phenomenon that happens when metal is exposed to oxygen and rusts. There are four iron atoms or hemopigments in one hemoglobin molecule. But in one little tiny erythrocyte, there's somewhere around 250 million hemoglobin proteins. So you can do all kinds of complicated math. But what this really means is that there is 1 billion iron atoms in one erythrocyte. And this means that one erythrocyte can bind to 1 billion oxygen molecules. Each oxygen molecule will eventually be used to make 36 ATP molecules. Doesn't matter which molecule you're really talking about, that's a lot of molecules. These are two great terms that neither you nor I use enough in this class. We should constantly be referring to these two terms when we think about whole blood or erythrocytes and the state of oxygenation that they're in. Deoxyhemoglobin is just one less hemoglobin protein that does not have an oxygen bound to its hemopigments. In this case, the hemopigment is very dark in color, giving the red blood cells a very dark brick red color. And this is because the reflection of melanin in skin is a dark brick red color in your veins, and it creates either cyanosis or pallor when it's not present. Cyanosis is the skin condition causing a blue tint or hue, and pallor is the skin condition causing a white hue to the skin. Both of these conditions are associated with someone who has low oxygen intake or low oxygen carrying ability. Deoxyhemoglobin is also found in the erythrocytes of all of the veins of your systemic circuit, but it's found in all of the arteries of your pulmonary circuit. These are the common places where deoxygenated blood exists. When I was in a foreign country, I used a bathroom in a train station, which I will never ever do again. But when I closed the door, a black light came on, and I was kind of like, "Is this a joke?" Not to mention, this was back in like late '90s, early 2000s. And I mentioned to some strangers that I was staying, well, not strangers, but to some people that I was staying with, and they said that it's because veins are harder to see in a black light. And that was to make it harder to do intravenous drugs in the train station bathroom. I thought that was kind of smart, actually. Oxyhemoglobin is simply just the opposite of deoxyhemoglobin. This would be a hemoglobin protein where all four iron atoms are bound to molecular oxygen. In this case, the molecule takes on a ruby red or a very bright, well, not bright, but a ruby red color. Oxyhemoglobin is found in the arteries of your systemic circuit and is found in the veins of your pulmonary circuit. If the blood vessels in your skin that are close to the surface become dilated, you can result in a skin condition called erythema. And this is normally called blushing, and it can be due to emotions, fever, heat, or variations in hormones.